Good afternoon, one and all. I welcome you all to Ganesh IAS Academy. In today's session, we'll be seeing science and technology based current affairs from the date February 13th to February 20th, 2024. Let us get into the news articles one by one. The first news that we are going to see is INSAT 3DS. What is this INSAT 3DS? Why is it in the news? Okay, so it is a satellite which has been recently launched by ISRO. So we need to get into the details of the satellite and the launch vehicle here. Let us understand it one by one. INSAT 3DS, this is the satellite. Okay, so the satellite INSAT 3DS on launch vehicle GSLV F-14, which is fully funded by Ministry of Earth Sciences. Important. Okay, and it was successfully launched by Indian Space Research Organization, that is ISRO, from Sriharikota, Satish Dhawan Space Center. Okay, so here the details about the satellite is important and the details about launch vehicle is also important. One more fact to be noted here is this is fully funded by Ministry of Earth Sciences. Okay, so let us understand the details of the satellite. So with the presently operational INSAT 3D and INSAT 3DR, which is in orbit, okay, they are orbiting satellites now. Now additionally INSAT 3DS is also added to it. Okay, so and this will augment the country's meteorological services like weather, climate and ocean related services. Okay, and the newly launched INSAT 3DS satellite aims to enhance the monitoring of the Earth's surface, atmosphere, oceans and environment, and it will also elevate the capabilities in data collection and dissemination. Moreover, satellite aided search and rescue services is also done using this satellite. Okay, so thus disaster management is also carried out with the help of this satellite. I mean with the help of the information that is shared by this satellite. Okay, now there are 4 different payloads that is present inside this satellite. What are the payloads? One is the imager, another is a sounder and a data relay transponder and satellite aided search and rescue transponder. Here you need to connect this point with this point. Okay, so how we are going to connect this? That it is going to enhance the monitoring. So for monitoring purposes, you need imager and a sounder, and it is also going to involve in data collection and dissemination, and for this process a data relay transponder is used. Moreover, a satellite aided search and rescue transponder is used because this application is there, that is satellite aided search and rescue service is there. Okay, so disaster management is also done. So for that we have another payload. So this is how you have to link the applications and the payload. Okay, what else do we have to understand about this satellite? That the data, the meteorological data from this satellite shall be utilized by various institutes of Ministry of Earth Sciences. There are different institutes like Indian Meteorological Department, National Center for Medium Range Weather Forecasting, Indian Institute of Tropical Meteorology, National Institute of Ocean Technology and Indian National Center for Ocean Information Services, INCOIS. Okay, and not only these agencies, it is not, I mean the information is not restricted only to these agencies but also other Indian agencies to enhance the meteorological research and services. Okay, and this will boost, I mean this initiative, this launch of satellite will boost India's weather and climate prediction and forecast, and it will also help or boost timely alerts and early warnings, and it will also give advisories for public and last mile users such as fisher folk and farmers. So for fisher folks, farmers and for other public, it will also give advisories at the times of danger. Okay, at the times of disaster. So these are all the information about the satellite. Now we need to know about the information of the launch vehicle. Okay, so what is the launch vehicle, what is its composition, all these details we need to know. So the launch vehicle here is GSLV F-14, which is a 3-stage launch vehicle. Okay, questions based on The number of stages have also been asked, so you need to give importance to these facts that it is a 3-stage launch vehicle. Here the first stage is a solid propellant motor and 4 earth storable propellant stages. So it has EPS. What is EPS? Earth storable propellant stages. What is it? It includes a supporting structure, propellant tanks and an engine. This setup is called as EPS, that is earth storable propellant stage. And the second stage, GS2, is also an earth storable propellant stage, which is important. But the third stage here is a cryogenic stage. Okay, so what is cryogenic? Gases that are liquefied, that are being used. Okay, gases at very low temperature is being used. So here the propellant loading of liquid oxygen and liquid hydrogen is used. Hydrogen and oxygen here are gases, but then they are liquefied. Okay, so only if we take them to very low temperatures, liquefaction is it is possible here. So liquid oxygen and liquid hydrogen is used in these cryogenic stages, 3 stages: solid, another EPS, another is cryogenic stage. And this point is also important, that GSLV 14 is the 16th flight of GSLV and the 10th flight with indigenous cryo stage. Why indigenous is important here? That is because earlier we were using this cryo stage which has been got from other countries like Russia, but this is indigenous. So this is the 10th flight with indigenous cryo stage, which is again important. So these are all the details that we need to know about this INSAT 3DS satellite, which has been launched recently with the help of the launch vehicle GSLV F14. Okay. The topic that we are going to discuss now is InfexnTM. What is InfexnTM? It is a next generation sequence testing. Who has developed this sequence testing? What is sequence testing? And all these details we need to know. Let us understand it. So Kerala's medical landscape is undergoing a great shift in the introduction with the introduction of InfexnTM. What is InfexnTM here? It is a revolutionary next generation sequencing test. And this will be India's first genomics based infectious disease test. So what all do we have to understand here? What is sequencing test and what is genomics based? What is genome? And these details we need to know. Okay, so it is designed to detect what will this do, what will this test do. It will detect any bacterial or fungal infection. So for detection of fungal and bacterial infection it is being used. Moreover, it is also used for it is also used for detection of relevant antimicrobial resistant genes. So not only bacterial and fungal infection, it is also going to detect antimicrobial resistance gene. This antimicrobial resistance is a huge problem now, okay, in medical field. So when we are able to detect this antimicrobial resistant gene, then it will help in the research, okay, and it can also be performed within the hospital settings. This is also important. This test can be performed within the hospital settings itself. We need not take the sample to any special laboratories and then wait for the sample, wait for the results for days, okay, but it can be done within the hospital settings. Moreover, this test represents a significant breakthrough in medical diagnostics and promises to deliver precise results within 24 hours. So this is also important. So it can give results within 24 hours and it can be done within hospital settings itself by utilizing the genome sequencing technology. So again, genome sequencing technology. What is genome? What is genome sequencing? And other details we need to know. And who has developed this InfexnTM? That is again important. It was developed by IIT Bombay based Haystack Analytics and Nurul Islam Institute of Medical Sciences, which is located at Nayatinkara, which is in Kerala. Okay. So this test kit will enable the effective implementation of national goal of championing antimicrobial leadership. So we have this national goal of championing antimicrobial leadership. As discussed earlier, this antimicrobial resistance is a main problem that is being faced by the medical field here. Okay, and it can empower the healthcare ecosystem with advanced definitive diagnostics, and this will ensure optimal antibiotic use. That is also important. So when we know which gene is responsible for antimicrobial resistance, and if we are able to address that particular gene, then the antibiotic usage can be optimized. Okay, so over exploitation of antibiotic usage can be avoided here. Next is, this innovative approach can enhance the accuracy and efficiency of infectious disease detection. That is also there. Now let us understand the background. What is genome, what is genomics, and other details. So what is genome here? Because this test, this InfexnTM, is going to do genome sequencing. Okay, so that is the reason why we are studying the background of what genome is. Okay, what is genome? It is the complete set of DNA instructions that is present in a cell. Very important. So here the keywords are complete set of DNA instruction. Complete set is important. Okay, now in upcoming slides we'll be differentiating this with gene. Gene is different and genome is different. Okay, let us understand the difference in a short while. Okay, so the human genome is made up of a tiny chromosome in the cell's mitochondria and 23 pairs of chromosomes that are found in the nucleus of the cell. So here the point to be noted is the DNA is not only present in the nucleus, but it is also, I mean, a tiny part of DNA is also present in the mitochondria. Okay, so mitochondrial DNA along with the 23 pairs of chromosomes that are found in the nucleus of the cells, together the complete set of information or the instruction is called as genome. Okay, so with the help of genome, so a genome contains all of the information that is required for a person to develop and function. So if we know the information about the genome of a particular person or a species, then we can know the complete detail of how they will develop and how they will function. Okay, so this can be understood with the help of understanding DNA. Okay, sorry, genome. And each DNA strand is made of 4 chemical units and they are called as nucleotide bases. What are the 4 nucleotide bases? All these are basic biology concepts. Okay, so the 4 bases are adenine, thymine, guanine, and cytosine. Okay, so that is how the bases are paired. Now if you see this in the picture, cell, nucleus, and inside nucleus we have nuclear DNA. Okay, chromosomes, I mean it is wound over histine, histamine. Next is mitochondria. Inside mitochondria you have mitochondrial DNA. So DNA is present in mitochondria, I mean a tiny part of chromosome is present here, and 23 pairs is found in the nucleus. Okay, so this plus this is genome. The complete instruction is genome. Next is, we saw about the nitrogenous bases, also called as nucleotide bases. So these are all the bases: adenine, thymine, guanine, and cytosine, and they are arranged in a particular sequence. We understanding this sequence is called as sequencing, which we'll understand in the next slide. So we already said that we'll differentiate what genome and what gene is. Okay, so what is gene here? It is a unit of DNA that contains the instructions of making a specific protein or a set of proteins. So gene is not the complete set of instruction, but it is the instruction for making a specific protein. Okay, so if I have DNA, if this is considered to be the complete DNA information or instruction that I have, if I take a particular part, this is gene. Okay, and what will this gene do? It will synthesize protein. Okay, but what is this? The entire thing is genome here. This is how you have to differentiate between what gene and genome is. So if I say that I have inherited certain gene from my parents, it means that I have inherited certain protein producing genes. So certain proteins will be produced. Okay, I'm not inheriting the entire genome, but then I'm inheriting particular gene from my parents. So this difference must be understood very clearly. Next is sequencing. What is sequencing here? Sequencing is the process of determining the exact order of the bases in strand of DNA, and this is called as sequencing. Okay, what is genome sequencing? It is the process of deciphering the order of the bases to decode the genetic fingerprint. This is important. Okay, so we are understanding the genetic fingerprint of a human with the help of genome sequencing. So what are all the steps involved in it? This is how it is done. An infected person is there, a sample from that person is taken, and then the targeted segment alone is taken and it is sent into the sequencing mission. Okay, and inside the sequencing mission it is compared with other known variants and then the sequencing is done. Okay, there will be different variants and then one variant is identified here. So this is how the sequencing process happens, and with the help of this InfexnTM, bacterial infection, fungal infection, and then antimicrobial resistance gene can be identified, and then it can be done within the hospital settings, and it can be, the results can be obtained within 24 hours. So these are all the important features of this InfexnTM, which has been developed by an institute in Kerala. Okay. The topic that we are going to discuss now is sodium catalyzed synthesis of carbon nanotubes. So here, why sodium catalyzed synthesis of carbon nanotubes is important, and what are all the applications of carbon nanotube, and what is the speciality with sodium catalyst. All these details we need to know. So sodium catalyzed synthesis of carbon nanotubes. What are carbon nanotubes? They are pivotal in advancing modern technology. That is because it showcases extraordinary properties and it finds applications in diverse fields like rechargeable batteries, flexible electronics, aerospace, transparent electrodes, touch screen, supercapacitors, and medicine. In all these fields, you can find the application of carbon nanotubes. But then the conventional CNT synthesis, that is CNT here is carbon nanotube. Okay, so the conventional CNT synthesis method require high temperature, that is approximately 1000 degrees Celsius, and a metal catalyst like iron, cobalt, and nickel. So metal catalyst is needed, and then high temperature is also needed. So these catalysts pose biocompatibility concern for potential biomedical application. Here we saw that it is finding application in medicine field, but then it has biocompatibility concern. That is because of the usage of these transition metals, that is iron, cobalt, and nickel. And here the challenge of removing these catalysts from the CNT adds a significant cost to the entire process, thus highlighting the urgent need for a cleaner and more sustainable CNT synthesis method. And this has been done recently. Okay, so who has taken the effort? It is the researchers of the Institute of Advanced Study in Science and Technology, which is an autonomous institute of the Department of Science and Technology of the Government of India, and they have pioneered a novel method for directly synthesizing the CNT on a glass substrate at temperatures of 750 degrees Celsius. So in the earlier process, we saw that the temperature was up to 1000 degrees Celsius, but here within 750 degrees Celsius, the process can be obtained, and this experiment is performed using plasma enhanced chemical vapor deposition technique. So vapor deposition technique is being used here, and then this innovative process circumvents the need for elevated temperatures as we discussed, and then it eliminates the necessity for transition metal catalyst also, because previously transition metal catalysts like iron, cobalt, and nickel were used. But now with this enhanced process, that is plasma enhanced chemical vapor deposition technique, there is no need for the usage of transition metal here. Okay, because when we use transition metal, the removal of the metal is being a difficult task here, which is adding to the cost of the process. Okay. So furthermore, this synthesis is executed under atmospheric pressure. That is also important. There, higher pressures are also needed, adding commendable cost effectiveness to its advantages. To its advantages compared to the counterparts in the field. The conventional methods that is used in the field. And among other elements, we have used sodium. Why? It emerges as the primary catalyst for initiating CNT growth in this new process. Okay, and it has also been observed that this sodium can be easily removed. How? By washing the sodium containing CNTs with deionized water. So just by washing the sodium containing CNTs, carbon nanotubes, with deionized water, these sodium metals can be removed. The metal particles can be removed here. But there, the removal process itself is a difficult task. Okay, so this is what we need to understand about the sodium catalyzed carbon nanotubes. The topic that we are going to discuss now is micellar water. What is micellar water? What are all its application and how does this micellar water work? Okay, so let us understand this. Micellar water, which is a product found in supermarkets, pharmacies and toiletries around the world, is commonly used to remove makeup. So what is its application? It is used to remove makeup and it is a very effective cleanser. Moreover, this micellar water typically contains purified water as its base. So the base here is purified water, thus making it gentle and suitable for various skin types, including sensitive skin. So this is its application. Its efficacy stems from the presence of micelle. This is where we are getting into the details, the science behind it. Okay, so it has something called as micelles, which is cluster of molecules with unique properties. So what is micelle? What is it made of? What are all the properties that is exhibited by micelle? This is important here. Okay, so with the help of micelle only, the micellar water functions. So these are the core components. What are the core components? The micelles are the core components here in the micellar water, and then they consist of the surfactant molecules which have both hydrophilic and lipophilic properties. This is important. So this is a surfactant molecule. So in a surfactant molecule, you have hydrophilic head and lipophilic tail. So what is hydrophilic? Hydro is water. Philic means attracting or loving. Okay, we have two words. This we would have studied in our chemistry, in our school chemistry, phobic and philic. What is phobic? Phobic means fear or I'm repelling it, and philic means I'm loving and attracting it. Okay, so hydrophilic means I'm attracting water, I'm loving water. Next is lipo, here means oil, dirt. Okay, so lipophilic means I'm attracting oil, but at the same time, what is lipophilic? Lipophilic is hydrophobic, I'm repelling water. Oil and water will repel each other, right? They won't sink. So it has hydrophobic tail and hydrophilic head, or otherwise hydrophilic head and lipophilic tail. Okay, so this is the property which is exhibited by one surfactant molecule, but this micelle is made up of many surfactant molecules. Okay, let us see that in the next slide. And this dual nature allows them to encapsulate and remove the dirt and oil from the skin. Okay, so because of this dual nature, the dirt will be attached to the lipophilic head and then it will get washed away in the water. Okay, this tail is lipophilic, which means I'm attracting oil. So it has this capability to attach to the oil that is present in the skin or the dirt that is present in the skin. So I'm attaching to dirt, and then when I wipe the skin with the help of these surfactant molecules and the micellar water, it will get removed. The dirt and oil will get removed. Okay, so yes, how does it work? working of the micellar water. So in a micelle, the hydrophilic heads of the surfactant molecules point outward. Okay, so the hydrophilic heads point outward. So okay, so each of these is a surfactant molecule and it forms micelle. Okay, interacting with the water. So the hydrophilic head interacts with the water while the hydrophobic tail remain tucked inside the micelle. Okay, so this is hydrophilic tail. It is tucked inside the micelle. Thus, it is staying away from water. Next is when the micellar water is applied to a cotton pad, what will happen? Okay, so this is a cotton pad. When I'm applying the micellar water to a cotton pad, a cotton bud. Okay, so when the micellar water is applied to a cotton pad, the hydrophilic heads adhere to the wet cotton. So the cotton is wet, water molecules are there, and this is hydrophilic, which means it is getting attracted to the water molecules. Okay, so the cotton wool will attract the heads of the surfactant molecules. Okay, thus exposing the hydrophobic tails, and then the hydrophobic tails are exposed. From this position, it will get into this position, the layers of lipophilic tails ready to dissolve oil and makeup. So the oilphilic or the lipophilic, oil loving tails are exposed here, and these tails are highly attracted to oil, makeup, sunscreen, dirt and other contaminants that are present in the skin. Okay, what else? So as the cotton pad moves across the skin, the hydrophobic tails of the micelle bind the oil and impurities, lifting them away from the skin surface. So this is what happens, and this is how it is used as a cleanser. So there are two different forms of micelle. One is reverse micelle, another is normal micelle. So in normal micelle, the hydrophobic tails are at the center. They are inside, they are not exposed, but hydrophilic heads are exposed. Okay, because it is going to interact with water. Whereas in reverse micelle, that is when it is involved in the process of cleansing, then the reverse happens. That is, here it is attached to the cotton wool. The hydrophilic heads are attached to the cotton wool, and then the lipophilic heads are exposed, thus it attracts oil. Okay, I hope it is clear. Micellar water. The topic that we are going to discuss now is rhodamine B. What is this rhodamine B, and why is this cotton candy making picture is shown here? Because it is linked to cotton candy making. Let us see why it is in the news. Recently, Tamil Nadu government has banned the sale and production of cotton candy in the state after it was found that the chemical rhodamine B was being used for making it. So, what is the problem in using rhodamine B for the production of cotton candy? That is because this chemical is a toxin here. Okay, so it is a chemical that is commonly used for dyeing in various industries like textile, paper, leather and paint industries, and it serves as a coloring agent, thus producing pink and red color. Okay, so this is the basic chemical structure of rhodamine B. And then it is a water soluble compound. This rhodamine B is a water soluble chemical, while it appears green in the powder form. So when it is in the powder form, it will appear green in color, but when it is mixed with water, it will turn into vivid fluorescent pink when it comes in contact with water. Okay, and as per the Food Safety Standards Act of 2006, preparation, packaging, importing, selling and serving food that are containing rhodamine B in any wedding ceremonies and other public events is a punishable offense. Okay, so rhodamine B is banned. Okay, and then since it is an industrial dye, it is not allowed in food anywhere in the world as it is toxic. This rhodamine B is toxic. You might have observed certain agarbattis and mosquito coils. which are pink in color. That pink color is actually from this rhodamine B. But then see, these people are adding this rhodamine B in cotton candy, which is consumable. Okay. Next is, researchers have identified that if food containing this chemical, that is rhodamine B, is consumed regularly, it can cause damage to the cerebellum tissue in the brain and also the brain stem that connects the brain and the spinal cord. So our brain function will be hindered if we are going to continuously consume this rhodamine B, and these damages can lead to functional abnormalities and can hinder human motor functions. Even our ability to move from one place to another will be hindered when we continuously consume this. And then the chemical is a toxic. It is a toxin, so it is toxic to humans and it can cause oxidative stress on cell and tissue if ingested. Oxidative stress will happen and it becomes particularly hazardous when it is mixed with food products, leading to cancer and tumor over time. So these are all the ill effects of consuming rhodamine B. That is the reason why recently Tamil Nadu government has banned rhodamine B from usage in cotton candies. Okay, so this is how you have to link one concept with another. Okay, so with this we have come to end of today's session. I hope you found the session to be very useful and informative. Let us see in the next session. Thank you all.